Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Desmosomes, which are also known as macula adherens. These are the major cell-to-cell adhesion complexes in the epidermis, bridging adjacent keratinocytes, thereby maintaining the epidermal integrity. These are made up of scaffolding of three major protein families. Number one, desmosomal cadherins, which contains desmoglein and desmocollins. Number two, armadillo family, which contains plakoglobin, plakofilin. Number three, Plakin family, which contains desmoplakin, NO and periplakins. These NO and periplakins are present in corneodesmosomes. These desmosomes are given this name because they are present in stratum corneum. That's why they are called corneodesmosomes. Here these three protein families are scaffolding proteins. That means each protein will go and anchor to another protein. That another protein will go and anchor to some other protein. Like that, first family will goes and binds to second family to third family. Finally, this third family, desmoplakin, is going and binding to keratin intermediate filaments, forming a contiguous network connecting adjacent keratinocytes with each other. Now coming to structure of desmosomes. Think this box as a keratinocyte and this portion is extracellular or intercellular portion where they form connecting bridges. Here from intracellular portion, our first family, which contains desmogleins, desmocollins, will protrude out because they are transmembrane proteins. That means which crosses the plasma membrane. Here you can see clearly this desmocollin and desmoglein is crossing the plasma membrane and entering this extracellular portion to go and bind to adjacent desmogleins and desmocollins, forming a bridge in this portion. If this bridge is broken in this portion, connection between two keratinocytes will be lost. That is nothing but acantholysis. Now clear, first family is transmembrane protein which crosses the membrane, and that crossed portion which is outside the cell is amino terminus of both desmogleins and desmocollins. Inside the cell is carboxy terminus. This carboxy terminus attaches to second family. What is second family? Plakoglobin, plakofilin, which in turn binds to third family. What is third family? Desmoplakin. Here desmoplakin also have amino terminus and carboxy terminus. Finally, with the carboxy terminus, desmoplakin binds to keratin intermediate filament. This carboxy terminus of desmoplakin is present in inner dense plaque. Amino terminus of desmosomal cadherins, that is desmoglein and desmocollin, is present in extracellular portion. Rest of all the proteins are present inside the outer dense plaque. This outer dense plaque is present nearest to plasma membrane, that is 10 to 20 nanometers. Inner dense plaque is present some more inside, that is away from the plasma membrane, that is 40 to 50 nanometers away from the plasma membrane. Clear? Structure of desmosome, we will see how it looks in diagram. This is a keratinocyte. This is its plasma membrane. First family is transmembrane protein family which crosses the plasma membrane. What are those? Desmoglein and desmocollin. Here you can see amino terminus is going outside the cell to form interconnecting bridges with the amino terminus of adjacent desmoglein, desmocollin, that then ultimately leading to formation of a bridge, intercellular bridge. Now carboxy terminus of first family binds to second family, plakoglobin, plakofilin. Second family goes and binds to third family, desmoplakin amino terminus. These all are present in outer dense plaque. Now we will go to inner dense plaque, that is inside, deep inside the cell, away from plasma membrane. which contains only carboxy terminus of third family, that is desmoplakin. Here you can see clearly carboxy terminus binding to keratin intermediate filament. Clear, first family contains two transmembrane proteins, desmocolin, desmoglein. I have clearly explained in dermoepidermal junction also, showing fingers downwards for two transmembrane proteins, that is BPAG2 alpha 6 beta 4 integrin. Here I am showing sideways, but desmosomes will be connecting all the layers of the epidermal cells in all directions, sidewise, up, down, connecting all the layers of epidermis with keratin intermediate filament. This keratin intermediate filament in the basal layer of the epidermis goes and binds to hemidesmosomes, which goes and binds to dermoepidermal junction via two another transmembrane proteins. Like this, they will form connection with epidermis, dermoepidermal junction, and dermis, maintaining epidermis and dermis tightly with each other, forming a contiguous network of structures, that is desmosomes, keratin intermediate filaments, hemidesmosomes, ultimately connecting to basement membrane zone and then dermis. Coming to applied aspects, if desmoglein 1 is destroyed either by genetic defects like in striate, type 1 striate PPK and Netherton syndrome, proteolysis, where this desmoglein 1 is the target of proteolysis by toxins released from bacteria like Staphylococcus scalded skin syndrome, bullous impetigo, and autoantibodies may also go and destroy desmoglein 1, leading to Pemphigus foliaceus, vulgaris, paraneoplastic Pemphigus, IgA Pemphigus, intraepidermal neutrophilic type. Desmocolin 1 is the target in subcorneal pustular dermatosis. It is also a type of IgA Pemphigus. Desmoglein 3, same IgA Pemphigus, intraepidermal neutrophilic type and paraneoplastic Pemphigus. This desmoglein 3 is the targeted molecular therapy for squamous cell carcinoma. Desmoplakin, envoplakin, periplakin, entire third protein family is destroyed in paraneoplastic Pemphigus. Here everywhere paraneoplastic Pemphigus is there, attacking all the proteins of all families of desmosomes. What is acantholysis? It is a process where loss of connection between keratinocytes due to breakdown of intercellular bridges, that is desmosomes. Then what happens if acantholysis occurs? Both keratinocytes will lose its connection and become acantholytic cell. It is of two types, primary, secondary. Primary is due to direct injury or via genetic defects in the structure of desmosome. So the primary pathogenesis is within the desmosome itself. Example, Pemphigus group, where autoantibodies are going and directly attacking the desmosome. Staphylococcus scalded skin syndrome, in which toxins will go and attack. Same, bullous impetigo also. Coming to secondary acantholysis. First, keratinocyte is damaged or altered. Then subsequently this interconnecting bridges, desmosomes, will get lysed. That is acantholysis. That is acantholysis secondary to alteration or damage in keratinocyte. Example, herpes zoster, herpes simplex and varicella. These intracellular viruses will damage the keratinocyte first, which may lead to subsequent damage to intercellular bridges, that is desmosomes. Coming to triggers. Autoantibodies, genetic defects, infections like Staphylococcus scalded skin syndrome, bullous impetigo, infestation like scabies, ionizing radiation, physical factors like friction. Friction will produce heat. Heat will produce sweating. If skin is continuously exposed to sweat, it may get macerated. Burns and trauma. All these physical factors are triggers for acantholysis. Finally, foods and drugs. Thiol containing foods like garlic, onion, drugs like penicillamine and captopril also. Then phenol containing foods like nuts, bananas, mango, tomato, drugs like phenobarbital, aspirin, rifampicin, cephalosporin. Finally, ACE inhibitors, glibenclamide also. All these foods and drugs we have to avoid for a pemphigus patient because they may trigger acantholysis. Clear? If you draw diagram and label by yourself, then only you will remember and you can draw in your exam. Now we will revise entire desmosome structure by labeling this diagram. Here you can see a keratinocyte. This box which is having plasma membrane. First family will cross plasma membrane because they are transmembrane proteins, desmoglein and desmocollin. They will have amino terminus, carboxy terminus. Amino terminus is extracellular portion. Carboxy terminus interbinds to second family, plakoglobin, plakophilin. Second family will go and bind to third family, which is nothing but desmoplakin. It will also have amino terminus, carboxy terminus. Carboxy terminus is within inner dense plaque, which goes and binds ultimately, finally to keratin intermediate filaments. Keratin intermediate filaments will go and bind to hemidesmosomes. Hemidesmosomes will go and bind to basement membrane zone, ultimately connecting epidermis with dermis. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.